My name is R. Dayanandan, Assistant Professor, Department of Chemistry, Sri Vidyamandir Arts and Science College, Katteri, Uttangarai. Today we are going to discuss about the topic nucleic acid. So what is nucleic acid? Nucleic acids are the important class of biomolecule found in all living cells in form of a nucleoprotein. நம்ம உடம்புல இருக்கக்கூடிய டிஎன்ஏ ஆர்என்ஏ டிஎன்ஏ இஸ் ஏ டிஆக்சி ரைபோநியூக்ளிக் ஆசிட் அண்ட் ஆர்என்ஏ ரைபோநியூக்ளிக் ஆசிட் இதை தான் வந்து நம்ம நியூக்ளிக் ஆசிட்ஸ் அப்படின்னு சொல்லி சொல்கிறோம். திஸ் நியூக்ளிக் ஆசிட் கண்டைன்ஸ் ப்ராஸ்தட்டிக் அண்ட் புரோட்டீன் குரூப். Prosthetic group is nothing but a non-proteinous part, that is the nucleic acid, DNA and RNA. And proteinous part, here we are not discussing about that proteinous part. We are taking the nucleic acid as a prosthetic group. So this nucleic acid are the genetic material of the cells responsible for the transmission of hereditary effect from one generation to another generation. Carry out the biosynthesis of protein. இது வந்து என்ன சொல்கிறோம் அப்படின்னா ஜெனட்டிக் கேரக்டர். நம்ம உடம்புல இருக்கக்கூடிய ப்ரோட்டீனஸ் பார்ட் அப்படியே இருக்கும். இந்த ப்ராஸ்தட்டிக் குரூப்னு சொல்லக்கூடிய டிஎன்ஏ அப்படின்னு பார்த்தோம்னா, ஃபார் எக்ஸாம்பிள் நம்ம வந்து சொல்லுவோம், ஒரு ஒரு குழந்தை பிறக்குது அப்படின்னா அந்த குழந்தைக்கு அந்த குழந்தையோட மூக்கு வந்து அவங்க தாத்தா மாதிரி இருக்குது அப்படின்னு சொல்லுவோம். அப்படின்னா என்ன மீனிங் அப்படின்னா, அந்த அவருடைய தாத்தாவுடைய டிஎன்ஏவும் அந்த குழந்தையுடைய டிஎன்ஏவும் எடுத்து நீங்கள் காரிலேட் பண்ணி பார்த்தீங்கன்னா 80% வந்து மேட்ச் ஆகும். ஸோ தட் ஹெரிடிட்டரி கேரக்டர்ஸ், த டிரான்ஸ்மிஷன் ஆஃப் ஹெரிடிட்டரி கேரக்டர், தட் இஸ் அ டிஎன்ஏ அண்டு ஆர்என்ஏ. ஸோ இதுதான் வந்து நம்ம சொல்கிறோம். நெக்ஸ்ட்டு இந்த ப்ரோட்டீன், இந்த ப்ரோட்டீனஸ் பார்ட் ஆல்ரெடி ஐ டோல்ட் யூ. The protein is nothing but the biopolymer of amino acids. There are 23 types of amino acids, in which 12 amino acids are essential amino acids and 11 amino acids are non-essential amino acids, which is synthesized in our body. மொத்தம் 23 அமினோ ஆசிட் இருக்குது. அதில் வந்து 12 அமினோ ஆசிட் வந்து நம்ம ஃபுட்டிலிருந்து எடுத்துக்கிறோம். மிச்சம் 11 அமினோ ஆசிட் வந்து நம்ம உடம்பே வந்து ப்ரிப்பேர் பண்ணிக்குது. ஸோ இந்த அமினோ ஆசிட்ஸினுடைய காம்பினேஷன் தான் வந்து நம்ம என்னன்னு சொல்கிறோம் அப்படின்னா புரோட்டீன், புரதம் அப்படின்னு சொல்கிறோம், which is found in our body. So nucleic acids is nothing but the biopolymer of nucleotide. So all nucleic acids can undergo stepwise degradation in 3 fundamental units. What are the 3 fundamental units அப்படின்னு சொன்னோம்னா, nitrogenous base, sugar and phosphate. Listen here. Each nucleotide, that is the nucleic acid on degradation, gives nucleotide. This nucleotide, if you break this nucleotide, you will get 3 parts. One is a sugar molecule, another one is a nitrogenous base, and another one is a phosphoric acid, H3PO3. You remember these 3, and first we will discuss one by one. First molecule, here you see the nucleic acid, DNA nucleic acid, which contains a base unit and a pentose sugar, that's a 5-membered ring sugar, and a phosphate group, which nucleotide on. Again, if you remove the phosphate group, you will get a nucleoside. Nucleotide is different from nucleoside. Okay, again, if you remove the base from the nucleoside இருந்து நம்ம பேஸை ரிமூவ் பண்ணோம் அப்படின்னா you will get a sugar and a base. So this from this we conclude that the nucleotide consists of nitrogenous base, sugar and phosphate. இந்த மூணுத்துடைய கலவை தான் வந்து நியூக்ளியோடைடு அப்படின்னு சொல்கிறோம். நைட்ரோஜினஸ் பேஸ், சுகர் அண்டு பாஸ்பேட், இதோடைய மூணுத்தினுடைய மிக்சர் தான் வந்து நியூக்ளியோடைடு. So first we will take sugar. What are sugars present in the nucleic acids? There are two types of sugars present in the nucleic acid. One is a ribose sugar and another one is a deoxyribose sugar. So these two sugars are pentose sugar. Pentose means it is a 5-membered ring. The ring contains 5 members. According to the nature of the sugar present, you see in ribose sugar, it contains 4 OH group. Whereas in 2 deoxyribose sugar, at the second position, the OH is absent. You listen this carefully. Here, in ribose sugar, at the second position, it contains OH group. In deoxyribose sugar, the OH group is absent. Next, the sugar component in RNA, it contains ribose sugar. And the sugar component in DNA is 2-deoxyribose sugar. So difference in between DNA and RNA is RNA contains ribose sugar and DNA contains 2-deoxyribose sugar. So ribonucleic acids are found mainly in cytoplasm of the cell, whereas deoxyribose sugar, that is DNA, are found mainly in cell nucleus of our body. Next part is first part is sugar that we have discussed, ribose sugar and deoxyribose sugar, and second part is a base. It's a nitrogenous base. So what are the base present in the nucleic acid? There are two types of base present in the nucleic acid. First type is a purine base and second one is a pyrimidine base. Purine base structure, which purine base contains one 6-membered ring combined with the imidazole ring, whereas pyrimidine contains a 6-membered ring which contains 2 nitrogen atom, whereas in purine base which contains 4 nitrogen atom. So this is the structure of purine and pyrimidine. There are two main bases, nitrogenous bases. You will see what are the purine bases and what are the pyrimidine bases. Purine bases are two types. One is adenine and another one is guanine. The structure of NH2 group varies in purine base. You see in purine derivative, it's adenine and guanine. So both adenine and guanine is found in both DNA and RNA. DNA, RNA ரெண்டுத்துலையுமே பார்த்தீங்கன்னா adenine and guanine present ஆயிருக்கும். That is a purine base. Whereas you take pyrimidine base, it is divided into 3 types. There are 3 types of pyrimidine base. One is a cytosine, another one is a thymine, and another one is a uracil. Here, cytosine pyrimidine base is found both in RNA and DNA, whereas thymine is present only in DNA but not in RNA. Whereas uracil, you take, it is found only in RNA but not in DNA. You have to remember this one. So there are 2 types of base, purine base and a pyrimidine base. Adenine and guanine are purine base, whereas cytosine, thymine and uracil are pyrimidine base. Here simply I have given in DNA, adenine, thymine, guanine and cytosine are present. Whereas in RNA, adenine, uracil, guanine and cytosine are present. You have to remember this one. And this nucleic acid on degradation, I have already told you, which gives a nucleoside. This nucleoside is made up of this base, that is nitrogenous base and sugar. This sugar is sugar and the base were linked with a beta linkage. Beta linkage in purine nucleoside, for example, the carbon 1 of the sugar is attached to nitrogen 9 of purine base. And in pyrimidine nucleoside, you see here, take this structure, adenosine. Here, the pentose sugar, that is ribose sugar, combined with adenosine, that is adenine. Adenine is the purine base combined with the pentose sugar through beta linkage. So adenine plus pentose sugar, the combination is called adenosine. So here the cysteine and deoxyribose sugar combination is called deoxycysteine. So it is a nucleoside. Here example of nucleoside. You take the purine base adenine combines with a ribose sugar, it is called adenosine. You take guanine, it combines with a ribose sugar called guanosine. When uracil combines with a ribose sugar, it's called uridine. When it combines with the deoxyribose sugar, it is called deoxyuridine. Similarly, adenine when combines with the deoxyribose sugar, it is called deoxyadenosine. You take cytosine, it combines with the ribose sugar called cytidine. Deoxyribose sugar, it is called deoxycytidine. Thymine, thymidine, deoxythymidine. So all the bases combines with the ribose sugar or deoxyribose sugar, and it was named according to the. You take nucleotides, which is composed of, already I told you, sugar, base and phosphate group. If you add a pentose sugar, nitrogenous base and a phosphate group, it is called a nucleoside. And if you remove the phosphate group, it is called nucleotide. It was given in that diagram. And also the nitrogenous base, here you see the nitrogenous base adenosine and thymine is linked with a hydrogen bonding. It's a weak van der Waals force of hydrogen bonding. Here cytosine and guanine is combined with a weak hydrogen bonding. So like this, and it also contains a phosphate group. Like this, a more number of combinations will occur, and we will see the types, that depending on the repeating units, there are so many repeating units like this. Depending upon the repeating units, there are two types of nucleic acid. Already we have discussed RNA ribonucleic acid, DNA deoxyribonucleic acid. The structure of nucleic acid. The primary structure of nucleic acid is the sequence of bases arranged in a strand. It is called as Western Crick model. Conclude that DNA was not a linear, it is a spiral, just like a spiral. It consists of two strands, linked with a spirally. And this Western Crick model of nucleic acid, which contains a sugar phosphate backbone on the outside and the bases on the inside. These bases on the inside contains hydrogen bonding. I already told you. The chains are held together by hydrogen bond between bases and on one strand and the bases on the other strand. And this is the structure of DNA. You see this here. The base thymine and adenine is combined with a hydrogen bonding, which contains a pentose sugar and a phosphate group. This pentose sugar and phosphate group and the nitrogenous base are linked with a beta linkage. And in second one, Guanine combined with cytosine. Again, thymine combined with adenine. Guanine combined with cytosine. Like this, the repeating unit goes on forming a double helix model. This structure is called as a Western Crick model or double helix model of DNA. That is a spiral model of DNA. This is the DNA structure. The DNA strands are not linear, I already told you, but it is a twisted or a helix model around the common axis. This helical structure is due to the presence of hydrogen bonding between the nitrogenous base. I already told you. If there was not, if you're not having any hydrogen bonding between the nitrogenous base means it will be a linear, but it was not linear due to the presence of hydrogen bonding. It has a double helix model. It's a twisted or helix model. The secondary structure is therefore known as a double helix model of DNA. So that's all about RNA and DNA, nucleic acids. I hope this class would be helpful for you. Thank you.